Hi guys, it's Kathy. I hope you're doing well. Just real quick, we have a live stream later today at 1 o'clock Eastern Standard Time. If you need to know what time that is for you, go and look at the live stream link. It's up and it'll tell you exactly how many hours until it's your turn. And then I won't be streaming tomorrow. There's no OJ tomorrow. So, but the link to the full OJ videos that we're watching are in every single description. So if you wanted to watch OJ tomorrow, you actually could just go and watch it. You could. It wouldn't be as fun. It would probably suck. But you could if you had to. Here we go. We're back with her again. We're going to be with her for a while, so I'm sure you're very shocked.
How many cycles you want it to go through.
In other words, this machine, you can program it or tell it basically how many of these cycles to go through, at what temperatures, and so forth.
That's right. And how long each cycle is to be. Like you're going to leave it at 95 degrees for 1 minute, and then you'll drop it down to 55 degrees and stay there for 30 seconds, and then you'll raise it up to 70 degrees and you'll stay there for 50 seconds or something like that.
This heating and cooling process that you described, was PCR, for instance, used before there were machines that did this in a fairly automated fashion?
It was, and my understanding was when PCR was developed that basically the people were doing this by hand. That is, holding the tube in a water bath at 100 degrees and then taking it out and putting it in another water bath at another temperature. So it would have been
Yay,
very
Kelly.
tedious to do
Thank you
that.
so much. Thank you.
You
Thank
have a lot of
you.
researchers standing around taking up a lot of their time holding their tubes in water baths. So anyway, the computer age came and this machine was developed to essentially take care of that for you. So you put your tubes in, you set the machine up, and turn it on, and it goes through as many cycles as you've programmed it to do, and then it holds your tube at a cool temperature until you come and retrieve it.
Just briefly, there's a step 3 listed that's called detection. Is it your understanding that we'll return to that in some detail in just a few moments?
Yes.
All right, with regard to this amplification process, and in particular how these strands are copied, would it aid you to use a prepared diagram showing 2 different double helixes or ladders?
That only really relates to the kind of detection that we're
All right, very
doing,
good.
so we could leave that for.
As far as the detection itself?
Yes.
Okay. Then if I could, I'd like to return to the drawing pad. And Dr. Cotton, could you use the diagram? And let's add a new page if we could, that I believe would be people's
249.
249. And with regard to this, if you could just illustrate, and I'd like you to actually write down a series of, just for illustrative purposes or example, bases, and how in this copying process, how the primer would then act to add these bases that are floating around in this tube mixture. Do you understand what I'm asking? Okay.
You want me to take a double strand, take it apart, and show how the second strand is.
Correct. Just one cycle's fine. And perhaps you could lay, what would be an appropriate title to illustrate that point?
More amplification.
Okay.
Oh, sorry, I didn't hear the
More amplification. It's going to take me a minute to set this up.
Very good.
Okay, so I've made a very short sequence, and I see there's a flaw in my thinking here, but we'll sort of try to work past that. Double-stranded, A's and T's, G's and C's, nicely paired up. Heat it, and create the two single strands. We're just unzipping the zipper. And now we want our primer to bind. So here's the flaw in my thinking. I'm going to have to make for a very short primer because I didn't draw a very long sequence. So let's say we have put in a primer here to have two T's, and we put in primer to fit the other side, which is going to be a T and a G. And you can see that this primer won't attach to this side, and this primer won't attach to this side, and the primers won't attach to each other. So you have to take these things into consideration. And then you simply have your enzyme come along, and the enzyme will come along and create this new strand. It will use this primer as its anchor point, and now add, and remember we've got, just individual A's, T's, G's, and C's floating around here available to be put into the new DNA strands. So the primer will come along and correctly make a brand new strand on both sides.
And what you,
And I said primer, but I meant polymerase. The polymerase will come along, hook onto this primer, and correctly finish off here the new strand.
so what you've illustrated is basically how this process works with the use of the primers and how from one strand you create two strands.
Yes,
And then that process just continues through the cycles, the number of times over and over that you described a few moments ago.
that's right.
All right. Then Dr. Cotton, if you would, and what I'm going to ask you is with excuse me, Army Admiral.
Again, why is there so much discussion about what she said? It's
Just a couple more questions if I could, Dr. Cotton. With regard to this primer, it latches on to the other side of the DNA strand, is that right?
It will bind, there is no other side. This is a single strand now, and this is a single strand at the point that you've heated it. So the primer is the first starting point to creating another side.
And it's from the primer on, up, or down the ladder, as the case may be, that it then starts this process of adding these bases one at a time.
That's right.
All right, very good. With regard to, and now just referring very briefly to the large chart, which I believe is People's Exhibit 248, as far as this, and you have described the second step on this chart, amplification, is that right?
That's right.
With regard to the third step, is that the final basic step in this process of PCR typing?
That's right. You're going to, now you've made lots of copies of your DNA, and you want to look at it. For purposes of forensic use, you will be copying sections of DNA that have some difference from person to person. And now you want to figure out, well, what does that difference look like for the sample that I just copied?
In other words, you actually obtain the types in a particular sample so that you can determine, for instance, what persons can be excluded or included as possible donors.
That's right.
With regard to this detection process, is it the same or different from what you're looking for using the RFLP method?
Depending on the genetic location that you've amplified, there is one of the detection methods is similar to the RFLP. That is, you're simply looking at how long a piece of DNA you amplified. You know you started at one end, but there's some variation in the length, and we may need to explain that a little bit more. But the other type of test that's available does not have anything to do with length, really, but has to do with a sequence difference. So that at a particular genetic location, I might have one sequence, Mr. Clark might have another, Mr. Goldman might have another. And so you could look at the sequence differences.
Do we have a board that will illustrate that difference as
Yes,
well?
we do.
All right, Your Honor, I'd ask that a prepared diagram be marked as,
Equals 250.
thank you.
Guess what I forgot? But it's okay because we've only been streaming for 12 minutes.
Your Honor, for the record, People's Exhibit 250 could be
There we
described as letter DNA
go.
with
That's
what appear to be
better.
two depictions of a ladder side by side, DNA ladder. All right. Dr. Cotton, with regard to this exhibit, first of all, have you had an opportunity to see it before?
Yes.
Would this exhibit help you describe the differences between these two methods of typing samples that you just made a brief reference to a moment ago?
Yes, it's the good example for, and it's a point of discussion for what is a sequence difference.
All right. Could you then use this diagram to discuss that?
Yes. What we have here is the same diagram basically that we were using yesterday to show what is a DNA molecule. And here we just have two of them that are side by side. And if you just go, let's say, from the top to the bottom, on one you'll see we have an AT pair at the top, and the other there's an AT pair at the top. Then we come down to a GC pair, and the second one also has a GC pair. But here in the middle, the DNA molecule on the left has an AT pair. DNA molecule on the right has a GC pair. So, and then if you go on down the remainder of the three base pairs here, they're alike, a GC, an AT, and another GC. So the only difference we have here is the fact that the third base pair down on one diagram is an AT and on the other is a GC. This is a DNA sequence difference. This sequence difference can be detected because you can amplify both of these pieces of DNA and then use a detection method to allow you to match either these pieces or those pieces, and you can figure out that you have two varieties basically in your sample. You have one molecule that has an AT pair and the other one that has a GC pair. And so in the PCR diagram that we were looking at a little while ago, when it got to the bottom and it says detection, this to amplify two pieces of DNA and then detect that they had a sequence difference is one type of detection that's used for forensic analysis.
What's the other type of detection method that the board just named very briefly?
The other type is looking at a length difference, which is very similar to what you're doing in RFLP. You're also looking at a length difference. But maybe we should go back to the piece of paper just very briefly.
And you're going to create a new diagram, Dr. Cotton?
Yes, I am.
All right. Your Honor, for the record, the witness is labeling this as PCR length difference.
All right, this will be 251.
What I'm going to do is go back very briefly to the same analogy or the same example that I used yesterday in terms of a repeat. So let's say we have the same CAT repeat, and I'm just using that because it's a very easy example. And I have two different DNA pieces here, and one has 2 repeats and the other has 4. And again, if I can somehow identify the ends of each of these pieces, I could look and see how long they were. For the RFLP test, we defined the ends by having an enzyme come along and literally cut these sections out. You can use the PCR test to also look at the lengths, and you can do that by designing your primers so they will come along and bind just outside the repeat. Now, we could go through and redraw all the amplifications, but if you think about, if we look at just one of the strands here, our primers come along and copy this section and copy this section. Ultimately, as you go through the PCR, you will end up with a piece.
I'm going to look and see if the defense steps in today.
That are this long. If you do the same thing with the other piece,
No,
your
not
primer
yet.
being just outside the repeats, the pieces that are eventually going to become your product or your many pieces of DNA that you've now copied will include the full length of the primers and these 4 repeats.
Not yet.
That I've diagrammed, whereas over here we only had 2.
Not this video.
Now, instead of having a sequence difference like the board that we just talked about, now you have PCR product, but the difference is a length difference. And you can analyze that length difference by separating the DNA strands on a gel. It doesn't happen to be exactly the same kind of gel we talked about yesterday, but the principle is exactly the same. And that is the DNA strands will move through the gel and separate out according to how long they are. And that is the other commonly used method of detection for looking at your PCR product, that is looking at some kind of genetic difference in the DNA that you've now amplified.
As far as these sequence differences, is my sequence difference at a particular set of markers different than yours?
It may be, and it may be the same. Since we haven't tested both of us, I couldn't answer that question.
What about in the area of these length differences?
The same thing. The variation in the population for these PCR markers is not extensive. So many of us may share some things, and then some of us would be different. So whether or not you and I are different, I couldn't tell you.
And that's
You
why.
may be, but I don't know.
Do you look at multiple genetic markers, more than one genetic marker, to determine these differences if they exist?
You look at as many genetic markers as you have the ability to look at, and no matter what kind of system you're using, be it PCR or RFLP, the more markers you look at, the more genetic locations you analyze, the more information you have. That's a generalization that will always be true for forensic testing. The more markers you look at, the more information you have.
And is that your goal as a forensic scientist, to look for as much information as possible?
Generally, that would be the goal, sure.
All right. I have, first of all, to your knowledge, do we have one more board to illustrate this PCR typing process?
Yes, I think so.
Okay. Your Honor, at this time, I'd ask to be marked as people's next in order
251.
a final PCR chart.
I'm sorry, 252, 251 being the drawing.
Your Honor, this is a
252.
252. It's also labeled PCR analysis, but it has 2 small blocks at the top, step 1, step 2, and then a large block, step 3, for the record. Now, Dr. Cotton, with regard to this chart, People's Exhibit 252, would this help you in describing how these actual differences, whether they're sequence differences or length differences, are determined by you as a forensic scientist?
Yes.
All right, then if you would, with this chart, then describe for the jury these differences in typing and how they work.
Okay, so step one, we extracted the DNA. Step two, we amplified it. We used the thermal cycler, like you saw in the previous diagram. And now, based on whatever genetic location we amplified, we're going to use one of these two types of detection. And the first type that's illustrated, which is on the left, is called a dot blot or sequence polymorphism. That is, sequence polymorphism just meaning sequence difference. It's exactly what we looked at on the two DNA molecules just a few minutes ago. So the setup is you have a nylon strip which has DNA bound to it in specific locations, and that's illustrated right here. There's a plastic tray, and the tray has like little wells in it, sort of, and the strips sit in the tray. Solution is added to those strips, and your amplified DNA is added to those strips. And the DNA that is spotted onto the strip will bind to whatever part of your amplified DNA that it matches up with. And where the DNA binds to the strip is then processed through a series of reactions to give you a blue dot on the strip where your sample DNA bound. And these blue dots are then interpreted to say you have, this is a term we haven't used. When I talk about genetic differences, if you go back to just mother and father, if you have two differences, those are referred to as an allele. An allele is simply a form of a gene. So that, for example, the two sequence DNA differences that we showed just a few minutes ago, those could be called two alleles, or the term Mr. Clark was using would be two types. So from the series of blue dots on the strip, you will read off the types or the alleles for that DNA sample that you just analyzed. And then you simply write those down and record those. The other method, which is diagrammed on the right.
I'm sorry, let me stop you for a moment, Dr. Cotton. And Your Honor, I'd ask that be marked as people's next in order what can be described as a tray containing various slots in it.
All right, 253, plastic tray.
Dr. Cotton, showing you what will be marked. I'm sorry, was that 253? You're on 253. What is that item you have in your hands?
The item is the typing tray. It's the tray that's illustrated in the top two pictures here. It's plastic. This tray has strips laid in the wells. The strips have not been processed, so there's no blue dots on them. And the tray has a clear plastic lid, which I taped down so that I wouldn't lose it. And this is the typing tray that's used in the laboratory, and examples of the strips that are used in the laboratory to go through this series of reactions of adding your amplified DNA and developing the blue dots on the strip. And from those blue dots, then you would read the types from those samples.
Where did that tray actually come from as well as, and I'm sorry, did you say there were strips inside the tray right now?
Yes, there are.
Where did they both come from?
They came from my lab, but they're manufactured by Perkin-Elmer Cetus, I believe.
Would that be a tray and strips then that you would have used in casework if it hadn't been brought to court today?
Oh, sure.
With regard to those strips, and they're currently laying in the bottom of the tray, is that right?
That's right.
What else is put into that tray other than the strips?
You want to go through the series of things that goes in here?
If you could, yes.
The strips go in first, and a solution that has some.
Did you see her face?
And
Like, that's a stupid question, sir. Why? Why are you asking me such a stupid question?
your amplified DNA goes in, and that's incubated in a water bath. So the water bath is controlling the temperature. The water isn't coming over the tray long enough for your amplified DNA to bind to the DNA dots that are on the nylon. Then a series of reagents or components are added which allow, well, first the extra DNA is washed off, and then a series of reagents are added to allow the development on the strip of the blue dot wherever your DNA bound. So you go through adding your amplified DNA, pouring off any that didn't bind, adding in a series the reagents that you need to develop the blue dots, pouring that off, and then allowing them to develop.
And then finally, do you see the appearance of these blue dots as is illustrated near the bottom left-hand side of People's Exhibit 252, the large diagram?
Yes.
All right, Your Honor, at this point, I'd ask that this exhibit be allowed to be distributed to the jury.
The plastic tray?
Yes.
All right, hand it to Dr. Cotton. Would you hand that to juror number 1, please? All right, Mr. Clark, would you collect 253 from Deputy Russell, please? And may I see that? Thank you. Doctor? Thank you. Mr. Clark?
Dr. Cotton, if you could you then turn to the right-hand side of People's Exhibit 252, and does that describe a different detection method or typing method when you're looking at these length differences?
Yes, it does.
All right. First of all, at the top, it seems to have some letters and numbers. What are those?
It reads AMFLIP D1S80 length polymorphism.
Okay, what's that first term mean?
The
AMFLIP.
AMPFLP is sort of a shorthand version of saying amplified length polymorphism. AMP referring to amplified, F is fragment, L is length, P is polymorphism. D1S80 is the nomenclature for the genetic location that is currently used in forensic labs that has a length polymorphism.
We'll return to that later, but is D1S80 simply a way of describing a particular genetic marker?
It's just the name of the marker.
Like PGM is the name of a particular
Exactly.
protein marker?
Exactly.
All right, go ahead.
Okay, so in this case, you have amplified DNA. The pieces that you amplified may have different lengths, and so you're using a gel, which is our green gel here with the red DNA on it, and the DNA is loaded into the gel. It's a slightly different type of gel than the one I described yesterday, but the principle of how the gel works is exactly the same. And the gel is subjected to electrophoresis, and the current moves the DNA through the gel. It moves through based on its size. And in this case, you don't have to use P32. You don't have to do anything fancy after you run the gel. You can simply use a stain and visualize where the DNA fragments are. So at the end, you have a gel that's stained, and you can actually see the DNA fragments directly, and then you can photograph that gel, and you can compare the positions of the DNA bands in the various samples that you've now analyzed.
And is that comparison of the bands like that same process of comparing bands using the RFLP method?
It is.
Now, with regard to this length difference and this typing method described on the right, is that again simply one of the means of typing a particular genetic marker following the use of this PCR copying process,
That's right.
as well as this dot blot or method used to look at differences between people where it's based on a sequence difference?
That's right. With the RFLP, everything you're looking at is length. When you use PCR, you can use a test that looks at sequence differences. That's illustrated on the left, or you can use a test that looks at length differences, and that's illustrated on the right.
One more question, if I could, and particularly while you're up, Dr. Cotton, I'd like to take you back to the current drawing, which I believe is People's Exhibit 251, labeled PCR length difference. With regard to that particular drawing, you described the use of these enzymes to cut the DNA at certain locations.
I simply made reference to that in that for PCR, scratch that, for RFLP to define the ends, that is to define the length, you use an enzyme to cut that length out. For PCR to define the length, to define the ends, you're using your primers. That defines the end, and then you're copying it, whatever, however many repeats happen to be in between. And whether you have a few repeats or a lot of repeats, that determines the length.
And these primers, how do they know exactly where to start this process?
The primers don't really know anything, but they are simply a piece of DNA that's single-stranded, and they are going in and binding with the piece of DNA in the reaction tube that matches them. That's why my short primer example wasn't very good, because to be very specific, the primers need to be longer, and they are usually around 20 or possibly more bases.
But for purposes of showing how this process works, that's why you used shorter lengths or shorter sequences.
Well, if we drew out all those number of bases, we'd be here all day.
We need a taller chart.
We would.
All right, very good. Your Honor, it was my intent to clear the charts and enter a slightly different area.
All right, proceed.
All right. Dr. Cotton, as far as the use of PCR in your laboratory, was there a time when you actually began using this technique in your actual casework?
Yes.
When did that happen?
We started doing PCR and casework in about, let me think about this a minute. I believe it was about June of 1992.
And you began using the RFLP process in casework when?
For forensic casework, we began about early 1988.
How do you decide, if a sample or a case comes into your laboratory, how do you decide whether to use this RFLP technique or to use the PCR copying process followed by typing at genetic markers that PCR looks at?
For our particular laboratory, that may be related to a lot of different things. For example, it may be that another lab had already done RFLP but doesn't have the capability of doing PCR, so they might specifically be asking us to do PCR. More typically, we would get a piece of evidence in, and we would have to make an assessment. Is this piece of evidence, does it have enough DNA, and is the DNA in sufficiently good condition to do RFLP? And if so, then you would proceed with RFLP. If the DNA or the piece of evidence contains only a very small amount of DNA, or the DNA is very degraded, then you would choose to go forward with PCR.
That's what I was just going to ask next. Do they each have their own advantages, these two different approaches?
Yes.
Can you describe that? Let's start with RFLP.
For RFLP, you need a larger amount of DNA, and it needs to be in very good condition.
Why is that?
Because the test, if you remember yesterday, I was talking about lengths of DNA that were thousands of base pairs long. You're looking at, in our lab, we're looking at a piece of DNA that's anywhere from about 1,500 up to 12,000 base pairs. So your DNA has to be in good condition it has to be much bigger than that as starting material, or your test won't work. So it has to be in good shape, and you have to have a fair amount of it. The RFLP test has the big advantage in that it's very discriminating from one person to the next. There's so much variation in the population that each time you look at another genetic locus or genetic location with RFLP, you're getting a lot of information. That is, you can discriminate one person from the next with a high level of discrimination.
Unfortunately, I do.
On the other hand,
but I only use them when I am too lazy to finish the dishes that day. I have one more favorite cup that's way bigger, but I'm using that for something else right now, so I can't really use it for coffee.
The PCR, the tests that are currently available, doesn't have that level of discrimination. It is a much lower level of discrimination, sort of in the range or maybe somewhat better than a whole series of serological markers. However, The PCR can use DNA when you have a very small amount, or it's in very poor condition, or both.
So in other words, in terms of the ability to tell many of us apart as human beings, in other words, a powerful test as far as this ability to, again, tell large populations apart, is the RFLP procedure then the more appropriate to be able to determine that type of information?
Absolutely. Then that would be the test of choice.
As far as the PCR process itself, and let's talk about both techniques in terms of how long they take to obtain results. Are there any differences between the two?
There can be significant differences.
Can you describe that, please?
If you brought a sample into the lab and you didn't have, say you didn't have anything else to do, every other case that you were working on was all finished, and you simply went through the PCR process, you could have results easily by the end of the week. So it'd be very fast. And that's assuming you have, a moderate number of samples, say 3 or 4 or something. Easily you would be done in a few days. The same test with RFLP could easily take you 3 months.
Why is that? Why does it take longer?
The length of the time it takes to do the RFLP test is pretty standard from the time you extract the DNA till you load it on the gel. But the exposure of the X-ray film to that nylon membrane, if you have a lot of DNA, it can be short as a day. But if you have little DNA, it can be as long as 2 weeks. So it could be that for the RFLP test, you took 2 weeks to look at one genetic location, then you took another 2 weeks to look at the second, and another 2 weeks to look at the third, and you can see that rapidly eats up a lot of time.
So it's that exposure or creating this X-ray film that takes a great deal of time, is that right?
It can, yes.
Are there differences in the time it may take to create this X-ray from a particular sample?
Yes, and the differences are basically related to how much DNA you have in your sample. If you think about a large amount of DNA and a small amount on your nylon membrane, you add your P32 DNA probe. If you have a lot of DNA there, you can bind a lot of probe. If you only have a small amount of DNA there, you only bind a small amount of probe. It's the emissions, the radioactive emissions from that probe that expose the X-ray film. So if you have a lot of probe there, you'll get an exposure on your X-ray film quickly. If you only have a little bit of probe bound because you only had a little bit of DNA to start with, it takes a long time to get enough exposure on that X-ray film to actually see something.
And again, if I can show you what was marked yesterday as People's Exhibit 246. Is that one of those X-ray films or autorads that can require this fairly lengthy period of time to develop?
Yes.
Now, you've described a little bit about the use of genetic markers, and you actually identified one of the particular markers. I believe it was D1S80, is that right?
That's right.
Is that just simply science's way of designating markers to tell one from another?
Basically, yes.
Is there any particular reason there's numbers and letters in it instead of just letters like PGM?
The DNA locations that do not code for, do not have information for a gene that we know of, are given letter designations. The D stands for DNA. The 1 is because that particular location is on chromosome 1. And the S80, I actually can't remember what that refers to, but that also has a specific reference. So, and they're called anonymous DNA pieces, that is. They're not a gene that we know about, and they're just given these D designations. It's an international nomenclature, so that if I read a journal article and it has one of these probes, it might say D5S20, and that would tell me that that DNA location was on chromosome 5. And if I could remember what the S20 meant,
Yay,
thank
thank you
As
so
far as
much.
these genes that
Thank
say
you so
have these
much.
statistics,
I
like
appreciate
starting
it.
with a D and
Yes.
the number of the chromosome and so on. You
That's
said
enough
there
time to have a
something about them
baby.
was not known. What is that?
Most of the, in fact, as far as I know, all of these genetic locations that contain
Thank you for the
these
bits.
repeats,
Thank you, Kelly.
these
Thank
are
you.
not genes in the traditional sense. Biologically speaking, in the traditional sense, a gene is a piece of DNA that contains information that creates a protein that then can go out into the cell and has a function. We know that these repeated sequences are not genes in that sense. Scientists don't actually know what the function of these repeats are. And that doesn't mean they don't have one. It
I have to know. Is it Maja? Maha? How do you pronounce that? Do you say the J?
just means we don't definitely know what it is.
Anonymous
As far
genes.
as forensic science, and in this process, do you have a process of deciding or selecting which genetic markers to look at for purposes of identifying people?
Yes, for all of the markers that are currently used, there's been a substantial development period. That is, of the markers that are available, people have gone in and done the kind of research to say how much variation is there in the population for this marker, how easy is this marker to use, how much development do we have to go through so that many laboratories could have access to this particular marker. So this kind of work is done in a development sort of stage, and then a marker is selected because of its utility, its ease of use, its discrimination power, and so on, to be then used in a wider setting with where many labs have access to or may choose to use a particular marker.
As a forensic scientist, would you, for instance, normally be interested in looking at the cystic fibrosis gene for purposes of your work?
No.
Why not?
Although there is a lot of variation in the cystic fibrosis gene, that variation isn't widespread enough in the population so that each time you used it, you would expect to get very much information. It wouldn't be sufficiently informative for forensic science purposes to use it.
So is it correct then that the genetic markers you're most interested in are those that show differences between people and can be easily used and are able to be typed, for instance, in older samples?
That would be right.
Now let's go back to PCR itself. Does it have uses outside forensic science, or is it just limited to work in an area such as yours, like human identification?
PCR is sort of the next thing that came along after the Southern blot that is used everywhere. The use of PCR in forensic science would be a very tiny fraction of its total utility to biological research.
What are some of the uses of PCR outside forensics?
PCR is used in the same kind, in genetic analysis, in gene mapping, and it's used in many basic research settings simply because it allows you to get a whole lot of DNA from a very small amount of starting material. And whenever you can get, scientifically, whenever you can get a lot of something, it gives you the ability to study it. So if you want to look at a particular section of DNA, then to get a lot of it makes your work a lot easier.
Now, you raised the term, or you used the term, genetic diagnosis. Is that determining whether an individual, for instance, either has a genetic disease or is, I think you used the term carrier?
Post more. More. Not less. Don't apologize.
Yes.
What is a carrier, just briefly?
Our
That is, if a
Discord.
parent
It's yours too.
has a genetic disorder, then the parent can transmit that genetic disorder to his or her children. And this is something that many parents want to be aware of before they start having children. And, so you might say, a family might decide, will we have children or will we adopt children? Because one or the other of the parents could be the carrier of a genetic problem.
Is PCR used, for example, to counsel parents, as you just described, about the likelihood of a child of theirs having a genetic disease?
Yes.
Is PCR used in the area of plant genetics?
I'm sure it is. I'm not a good enough plant biologist to know any specific applications, but I have no doubt that it's being used.
You used the term gene mapping. Can you just tell us a little bit about that?
In terms of understanding human diseases and human genetics, one of the primary things that scientists need to do or want to understand is how are genes located relative to each other? What gene is on what chromosome? What genes reside close to it? And so in the process of answering these questions for specific genes, PCR would be a very helpful technique.
Is PCR used, for instance, in the identification of the remains of American war dead?
Yes.
All right, ladies and gentlemen, we're going to use this point to take a brief court reporter recess for the morning. Please remember all my admonitions to you. Don't discuss the case amongst yourselves, form any opinions about the case, conduct any deliberations until the matter has been submitted to you, or allow anybody to communicate with you. And we will resume at 10 minutes till 11. All right, Dr. Cotton, you can step down.
Thank you, Your Honor. Dr. Cotton, you've described a little bit about the use of these tests, whether RFLP or PCR, to exclude people or include people. Is that right?
That's right.
Is there any difference between those two things, excluding someone or including someone?
Well, those are two entirely different things, but I'm not sure what you mean.
All right. Well, first of all, in excluding a person, what does that mean once you've conducted the test?
You're saying that person cannot be a contributor to the sample that you've tested.
What about the opposite? What about including someone?
Including someone, you're saying that this person could be a contributor to the sample that you've tested.
Is there, in terms of the use of these tests, are they capable and do they in fact serve both of those purposes?
Yes, they do.
Now, as far as the use of PCR, and you've described the fact that there are genetic markers or various locations on this DNA molecule that you look at where people differ,
That's right.
And you described a little bit about the selection process, the process whereby forensic science decides which markers to look at. Is that right?
That's right.
Are there specific genetic markers that you look at in your laboratory following PCR amplification?
Of course.
Okay, do they have names that describe them?
Yes, they do.
Could you describe those for the jury, please?
To tell you the truth, so I don't make a mistake, if you want me to list those markers, how about if you hand me one of my notebooks so I
Sure.
can read off of that?
Would that assist you in describing
think it
each one
would.
exactly?
Yes.
First of all, I'm sorry. First of all, Dr. Cotton, these what appear to be two binders. Can you just tell us what they are?
The two binders have copies in them of our original case folder. I have the original case folder in my briefcase, but these are easier to manipulate.
In other words, those are easier for you to work with?
That's right.
Actually, let me approach it slightly differently. Is there a marker that you examine using the PCR process called DQ alpha?
Yes.
What is DQ alpha?
DQ alpha is located on chromosome 6, and it is one of the human leukocyte antigen markers. These microphones are doing funny things.
Okay. Now you used a fairly large term, human leukocyte antigen.
That is the name, generic name of the set of genes for which DQ alpha is one. They are surface, they code for proteins that are on the surface of cells. And DQ alpha is one of these proteins that's been very well characterized. And the DNA that encodes this protein.
Let me look
is
around real
also very
quick.
well
1
characterized.
second.
Dr. Cotton, could you pull that microphone just a little closer to you?
Okay.
Did you disconnect the court's microphone? Take a recess. Fix it.
Of course, as soon as I go to look, this happens. This as soon as I go to do something, Judge Ito steps off the bench and makes me come over here and fast forward it. And now they're going to have a sidebar. You
Please
watch.
proceed. Mr. Clark.
Thank you, Your Honor. Dr. Cotton, I believe you were about to discuss this marker called DQ alpha.
I can't.
That's right.
And is that one of the
Law and Network is copyrighted. So I can play, I could get, like, I can't, no, it's complicated. So I won't get anything off Law and Network because it's too complicated to be able to play it on my channel. But yeah, that's what I'm looking. I'm looking to see if I can find it somewhere else.
markers that you typed samples at following the use of this PCR copying process?
Yes, we did.
What can you tell us about DQ alpha? How well does it tell people apart?
DQ alpha has 6 alleles. That is, there's 6 different forms of this DQ alpha gene. And with that many alleles or forms, you can generate 21 different types in people. That is, so you're not confused, we each have 2 forms of any genetic locus or any gene. One comes from mother, one comes from father. So you're looking at how many combinations of these 6 things would exist, and there are 21 different combinations of the 6 alleles for this gene, DQ alpha.
Is there a particular term used in science to describe what a person's type is when they have 2 forms of a particular genetic marker?
Yes.
What's that called?
I mean, what I think you're talking about are the terms homozygous and heterozygous.
All right, well, let's start with that first. What do those terms mean? Are they important? Do we need to know those terms?
Do you need to know? Well, I don't know if you need to know them. Probably you do. When you have inherited from both parents the same form of a gene, that is, so both your copy that you got from your mother and the copy you got from your father are identical, then
Welcome.
you would be said to be homozygous. Homo meaning the same.
Hope Phillips,
and
a
zygous
trainer
referring
and
to
curriculum.
zygote, which is combination of egg and sperm. So if you have 2 forms, you will always have 2 forms of the gene, and if they happen to be the same, let's use a very easy example. If you got a gene for blood group A from your mother and you got a gene for blood group A from your father, you would have two genes, both are for blood group A, and you would be homozygous for that A.
All right. Is there also a term called genotype?
Genotype simply, it just means what types do you have? Do you have an A and an A or an A and a B? That's your genotype, AA or AB.
As far as this marker DQ alpha is concerned, then if everyone in the courtroom were tested, would each of us be one of these 21 different types or genotypes?
We would each be one of those 21 types.
Does that mean, because there are presumably there are more than 21 people in this courtroom, does that mean that it's likely or necessarily true that at least
I might be able to get it, but I doubt it. So they do stream live streams, but they don't stream of court cases. They stream their committee meetings. But what that means is they have the system in place to stream every day if they want to. And because this trial is so big, they might. So I turned on my subscriptions and my notification bell for there. And if it pops up, I can get it. If it doesn't pop up, then I can't get it from Law and Crime.
two of us have the same genotype?
Well, probably for those genotypes or combinations of genes that are common, we'd find more than one person in the courtroom who had the same type.
That was actually going to be my next question. As far as these types that we each have, are they distributed absolutely equally or not?
They're not.
Why is that?
I don't think we can. First of all, I'm not sure I can answer that question. That may be an evolutionary answer, and I'm not that knowledgeable about the DQ alpha gene. However, what I know is that the types are not distributed evenly. So some types are very rare, some types are relatively rare, and some types are very common.
We'll return to it later, but are there studies done to determine how common or how rare these individual types are?
There actually is a very large amount of work published on how common or rare particular DQ alpha types are in many different racial or ethnic groups.
Still with this DQ alpha marker, how do you know it's a good marker to use in forensics?
Well, if you use as your judge the first criteria that you want something to be relatively variable, that is, a marker that shows a lot of variation is more useful than a marker that doesn't show very much variation. So on that criteria, DQ alpha is quite good because with 6 alleles giving you 21 different types, that's a fair amount of variation and it's going to have useful information. The test that has been designed for forensic use for DQ alpha, which uses PCR, only amplifies a very short section of DNA. That means the test is going to work well on samples that are degraded. And so for that reason, it's also very good.
Is it, for instance, more useful than the ABO blood grouping types that this jury has already heard about?
Yes, it would be.
Why is it more useful?
It has more variation.
In what way?
Well, with ABO blood groups, you have 3 choices of alleles. You can be an A, you can be O, or you can be B. And that gives you a combination of 6 different genotypes, that is, taking those 3 things 2 at a time. So that means each one of us will be one of those 6 types, as opposed to DQ alpha, where each one of us would be one of these 21 types. So DQ alpha is going to be able to distinguish us one from the other better than ABO.
Incidentally, of these 6 types for ABO you've described, are they actually fewer than that can be actually determined from testing?
With normal serology testing, you can't distinguish all those types. People are developing DNA tests in which you will be able to distinguish all of those types.
All right. In addition to DQ alpha, do you type other genetic markers following the use of PCR?
Yes.
And are they collected? First of all, how many other markers do you type in the laboratory?
In our laboratory, we're typing 5 other markers. Actually, recently we've added 3 more, but they were not done here and we don't need to worry about those. So for purposes of this case, we typed 5 additional markers besides the DQ alpha.
Are those 5 markers collectively known by any term?
The 5 markers are collectively referred to as polymarker or PM, and that's simply the name given to that's not a scientific name, really. It's just the company who developed this test refers to it as PM, generally standing for polymarker.
And are there different ways of describing those 5 different markers that are part of the polymarker system? In other words, are they labeled by a particular name?
Yes, of the 5 genetic locations that are tested in this polymarker system, each one has a specific name.
And can you briefly describe what each of those 5 are in terms of their name?
Sure, but this is what I'm going to take out of my notebook here.
All right.
So.
All right, Mr. Neufeld, do you want to see that?
I'm just going to read this section here. That I can do too.
All right, Mr. Clark, proceed.
Yes, thank you. Dr. Cotton, what are those five genetic markers?
Each one of them has a short abbreviation, which stands for the longer name. If you just focus on the abbreviation, that's probably a lot easier. The 5 markers are LDLR, standing for low density lipoprotein receptor.
Can we stick with LDLR?
You want me to just give the initials?
That's fine.
Okay. The other is GYPA. The third is HBGG. The fourth is D7S8. And the fifth is GC.
Now, of those 5 you just described, one of them had that notation of the letter D with a number. Is that right?
That's right.
Would that then be another one of those markers where its function in the body is unknown?
As far as I know, it is, yes.
And then the other 4 were described by letters, basically. Is that right?
The other 4 are actually genes, and they have the letters are just an abbreviation for the name of the protein that's been very well described for that gene.
Now, this set of 5 markers that are part of polymarker, how do you know they're good to be used or appropriate to be used in forensic work?
Each one of these markers does not have a lot of variation, but grouped together, it still is informative. And really the answer to your question is, in addition to the theoretical idea of how informative is this marker, this test has been designed to be also useful specifically for forensic purposes. The sections of DNA that are amplified are small, and the typing is on the dot blot strips that you saw on the tray a while ago. So it's a test that's easy to do in the laboratory, and it works on and has been shown to work on a very wide variety of samples. So it's applicable to all kinds of samples that come in terms of forensic casework.
Incidentally, that tray with the strips that you identified and was shown to the jury, was that actually a polymarker set of typing strips?
Yes, the strips that were in the tray when you looked at them have the 5 polymarker loci, and if you were looking at the strip, you would see that there were letters on the top of each one, and it's the LDLR, GYPA, indicating the dots in that set of this in that portion of the strip are for that particular genetic location.
All right, referring to equals 253.
Yes, thank you, Your Honor. Going back to DQ alpha for a moment, how long has it been used in forensic casework?
I think since about 1986.
Going forward then to the polymarker genetic markers, when were they begun to be used in forensic casework?
We started using the polymarker test in January of 1994. There probably are some other labs who were using it earlier than we do, but I don't know exactly how. It wouldn't have been a lot earlier, somewhere maybe within a year or so before we did. So let's say it's been used in forensic casework since approximately 1993.
Is there some process that these genetic markers undergo before they're actually used in casework, or does someone just discover a gene and start using it immediately?
There's actually what usually ends up being a very long process of development of the marker, and then what's referred to as validation. That is, if another laboratory uses a marker, that doesn't necessarily mean that I can just bring it into my lab and start using it. So each lab that uses a particular test generally goes through a set of experiments in their laboratory to make sure that in the hands of the people in that laboratory, the test functions as it's supposed to. It meets its specifications, and that everybody that's using it understands what it is they're doing. So there's development of the marker, then there's validation of the marker, which may occur in many different labs, and then there's training.
I did not. It just confuses people when I do that. They get really confused, and they wonder where the stream is, and then they're like, "Oh, this is 18 hours from now." So no, I will when it gets closer, but no, I did not.
The staff in a particular lab to use that marker.
And do you ensure that that marker can be correctly typed in evidence type samples before you even begin casework use of them?
Of course.
As far as these polymarker, well, let's start with DQ alpha. Is DQ alpha a marker that's tested? And I think you said more than one laboratory uses the polymarker system. What about the DQ alpha system?
The same is true of the DQ alpha system. It's used by many forensic laboratories around the country and really around the world.
That was going to be my next question. To your knowledge, are the polymarker and DQ alpha genetic markers used worldwide?
They are.
You touched a little bit about, as far as the 5 polymarker genetic markers, that their variation is not as great as DQ alpha.
That's right.
Without getting into each one individually, can you tell us briefly how much variation they each show?
The 5 locations that make up the polymarker system, all of those 5 locations either have 2 or 3 alleles or forms, and they all are designated A or B, and if they have 3 forms in the kit, they're designated A, B, or C. So you can, for example, if you have a marker that has an A type and a B type, any given individual then could have 2 A's, they would be an AA. They could have 2 B's, they would be a BB, or they could have an A and a B, and they would be an AB. So if you have 2 alleles, the most number of genotypes you can have is 3.
DQ alpha and these polymarker genetic markers, do they represent sequence differences or length differences as you described them earlier today?
These are all sequence differences.
As far as the actual typing of these DQ alpha and polymarker genetic markers, do you actually conduct these tests from scratch in terms of materials and what you use to be able to type samples?
No.
How do you do it?
These tests, the DQ alpha and the polymarker, come as a kit, which means you're buying in a box all the things that you need, or almost all the things you need to do the test. You'd end up making a few of solutions in the laboratory, but the strips come in the kit. The chemicals that you need to do the reaction to create the blue dots come in the kit. The tray comes, you have to buy the tray and the kit. I guess actually you buy them separately. And the PCR mix that contains the primers and the polymerase and the A's, G's, T's, and C's. All that comes in the kit. So you're just supplying some very simple to make solutions and using the kit to do the test.
When you conduct the more powerful RFLP typing technique, do you use kits to do that?
No.
Why is there a difference between the 2 approaches to DNA typing as far as kits are concerned?
Well, somebody took the time to make the kit for DQ alpha and polymarker, and nobody has tried to market a kit for RFLP testing. I guess you could, but it would have an awful lot of components.
Is there anything unusual about using kits to conduct testing?
Oh, no,
Why do you use a kit?
because it's there.
Does it have
It's a good test, and it's available, and it's had an enormous amount of work done on it. If it wasn't a good test, you wouldn't use it. If it wasn't available, obviously, you couldn't use it. So the company that makes the kit has put a lot of work into it, and it happens to be very, very good.
All right, turning your attention, if I can, to the last genetic marker that we'll talk about, D1S80. Does that represent a genetic marker that's typed based on length differences as opposed to sequence differences?
Yes.
Was D1S80 developed as a marker to be used in forensics, or did it have some different origin?
As far as I know, it wasn't discovered. I mean, it was discovered, but then basically it was developed specifically for forensic use. I really don't know if there's any research use for D1S80 or not.
Outside forensics?
Right.
How are you familiar with this particular genetic marker?
We've run this marker in our lab. We're not currently using it in our routine casework, but we've run it in our lab. And I've also actually read a number of papers that had to do with D1S80 and heard a lot about it at various scientific meetings.
Why don't you use it in your casework in the laboratory?
The reason we're not using it in our casework is not because it's not a good marker. It's a very practical reason, and that is the kind of gel that the D1S80 is run on is not a kind of gel that we would run other markers on. And so essentially we aren't using it because it's a fair amount of work to do it, and you get one piece of information off that is the D1S80 piece of information. There are some other markers out there that we chose to use instead. They're not quite as informative as D1S80, but I can do more of them at a time. And so it was a very practical decision that you would just make in the lab to say, do we want to do test A or test B? And has nothing to do with whether or not D1S80 is a good marker. It was just our choice to go with another type of marker.
So there are practical considerations that go into each laboratory's decision of what markers they're going to test?
That's right. And because not every laboratory that does casework has the same type of casework or the same casework demands. And so all of those things go into your decision about what markers you're going to be using.
As far as your familiarity with this D1S80 marker, what type of variation does it show?
The D1S80 has over at least 24 alleles, that is at least 24 forms. I haven't calculated how many genotypes that could give you, but you can imagine at least 24 things taken two at a time. You can have many, many, many combinations. So it's a very informative marker, and it's, except for the gel system, which takes some pains to do, it's a very good one.
All right, I'd like to shift topics if we can and ask you, are you familiar with the term contamination?
Certainly.
What does that term mean to you?
It means so many different things that maybe I should try to break them down in
All
terms of what it means to me. If you think of a biological sample, say you take a blood sample from someone in sterile conditions, you have a very clean, sterile sample. That's sort of the ultimate in terms of having a good sample. As soon as you take that sample out of a sterile condition and say you have a blood stain on a piece of cloth, then you could think about, well, that blood stain is no longer sterile and it could be, quote, contaminated by anything that's on that cloth. So that would sort of be a second, or let's say a first level. Now, in terms of analyzing evidence from a crime scene, you're always getting things that are like that. You're never being presented with a sterile sample. So in terms of a crime scene, contamination would mean that the sample is that is, in retrieving the sample, it is as it was deposited, that nothing's been added to that sample in the process of picking it up, taking it to the crime lab, and so forth. And then you can also say, well, I have my sample from the crime scene. It came to the crime lab in exactly the same condition that it was at the crime scene. And then clearly you need to be concerned with whether or not in the process of working with that sample in the laboratory, are you introducing any contamination from any source in the laboratory? So you can't use it very well as a generic term. I mean, you could, but unless you specify what kind of contamination you're talking about, then it becomes very difficult to discuss, because you can break it down and you can have many different kinds of contamination, some of which are inherent in the fact that you have a piece of evidence and not a sterile blood sample, and some of which are not inherent and that could occur along the way in how that sample is handled.
As a forensic scientist, are you concerned about contamination?
Of course.
Why?
The idea of analyzing a piece of evidence is that you learn something about that piece of evidence. You're not interested in learning about, that is, if you contaminate that piece of evidence with, let's talk about, say you contaminate a piece of evidence with some other biological sample or some DNA in the laboratory, that will then interfere with your being able to come to a valid conclusion about that piece of evidence. And so you would certainly prefer not to have anything interfere with coming to a valid conclusion about a specific piece of evidence.
As far as this area of contamination, and you've described the two basic DNA typing approaches that you use, both RFLP and PCR, is your concern about contamination the same for both of those techniques, or is it different?
It's different.
Why?
The PCR test is much more sensitive. You can do a PCR test with a very minute amount of starting material. So you would be more concerned. It doesn't mean you're not concerned with contamination for RFLP, but you're going to be more concerned with contamination when you're dealing with doing a PCR test on a piece of evidence.
Okay, let's take RFLP to start with. What steps, if any, do you take to deal with the problem or the potential problem of contamination in that testing process?
Okay, and my answer is going to then be what we're doing in the laboratory, because we're simply receiving a piece of evidence from outside. That is, we're not collecting the evidence ourselves. In the laboratory, our work is done in it's called a biological safety cabinet, but basically it's a setup where you have a piece of glass and you have a working area, and the air is circulated within that working area to both protect what you're working on and to protect yourself. So you have some containment in the area that you're working with these samples. In our laboratory, when a sample is moved from one tube to another, that is, the tube has a label, it has a case number label and a sample number label, and in the process of doing DNA extractions and doing this testing, there are points where you have to remove the sample from one tube and put it into another. That transfer, the labeling on the tubes is witnessed. That is, somebody's checking to make sure that if I'm transferring from a tube that's marked 02, then I'm transferring it. The second tube I have in my hand is also marked 02. The other main precaution is that we do the DNA extractions for evidence samples as opposed to known samples. That is, when I say known, I mean you have a standard sample from a known individual. Those extractions are not done out in, well, they're done in this hood, but they're not done at the same time. So the knowns and the evidence samples are not worked on for purposes of DNA extraction at the same time.
So those are the primary steps undertaken with regard to the potential for contamination as far as RFLP typing is concerned?
That's right.
What about PCR? Do you undertake any different steps or additional steps to deal with this potential problem?
There are many additional features of how you do a PCR test to deal with minimizing any problems with contamination.
Could you describe those? And are there general categories that you can place them into as far as the steps that you take?
I'm not sure what you're asking.
Well, what are the steps you take to deal with this potential problem?
Okay. The
Someone just needs to tell him he could raise more money for his 2026 campaign if he was streaming.
Let me think about this a second so it comes out organized. The PCR, the DNA extractions for PCR are done in a separate location than the extractions for RFLP. Now,
No.
in our lab, it happens to be that there's a separate.
No, you have to come by horse. You're only allowed to come if you're riding in on a horse.
One of these
And the
biological
horse
safety
you
cabinets
rode in
that's only
on,
used for
sir.
PCR extractions. And the reason for that is that if you're doing something, if you have enough DNA to work with RFLP, that's a fair amount of DNA compared to what you might have in a PCR sample. So we don't want to be handling the large quantities of DNA that you would have for RFLP in the same place with the same equipment that you're handling the very small amounts. So most laboratories will have a separate location to do DNA extractions when those DNA extractions are for PCR. And in our lab, it's a separate one of these hoods. The sample is, and in addition to that, for example, in the hood, the pipetteman or the piece of equipment you're using to add things to the sample and take things out of the sample are specified. That is, you have a pipetteman that's used only for DNA, and you have a series of pipettemans that are used for everything else, so that you're not interchanging the piece of equipment that you're using to handle the DNA. You are that concerned. And the tips that you're using for the pipetteman are these art tips that have a filter in them, so that you can't get any part of your sample up into the pipetteman and then therefore accidentally transfer it to the next thing that you work with.
Now, I believe, did you talk about extraction? I'm sorry, extracting evidence?
Well, basically that's everything I just said relates to the area where the DNA is extracted.
All right. Extraction was the first basic phase of dealing with evidence for purposes of PCR typing. Is that correct?
That's right.
Earlier, you described the second major phase involves what's called amplification, this copying process.
That's right.
Do you take any precautions or additional steps to, again, deal with the potential for contamination during this particular phase?
Yes.
What are those?
Well, first, now that you finish the extraction, you're going to set up the amplification. That is, you go through that process where you have a tube, and in that tube you put your DNA and you put all the things you need to do the PCR, the primers and the polymerase and so on. That setup is done in a second area, a very clean area, and it's in our lab. It happens to be a very small room, but it could be some other just separate location. So the setup of the PCR reaction is done away from where the DNA extraction is done. Once that reaction is set up, it's then carried to yet a third location where the thermal cycler is, where the amplification takes place. Once it's gone into that area where the amplification takes place, it does not ever go back in the other direction.
Why is that?
Think about, in terms of the PCR, you have started with a tiny amount of material, and now, after the process of amplification, you've generated millions of copies of that tiny amount of material. The biggest contamination problem that can occur with PCR is transferring some of that amplified product back to where you're starting out with, because that amplified product will amplify really well if it gets into anything else. So it's not the only concern, but the biggest contamination concern is that you do not get any of that amplified product, which is now a lot of DNA, back into anything that you're ever starting out with. So the DNA samples go from the extraction area to the setup area to the area where the thermal cycler is, and where you then do the analysis of the amplified product, and they do not go back in the other direction.
And that's to, again, avoid this problem of lots of DNA being mixed in or commingled with small amounts of DNA.
That's right.
Now, as far as, and I believe you described the fact that evidence is not extracted at the same time as known samples, or did you mention that?
I did mention that with regard to RFLP testing, and that same general procedure, that is, you don't do the DNA extractions for evidence and knowns at the same time. That's just a general precaution we use in our laboratory, and it's applied to everything.
So as far as RFLP typing, your primary steps to avoid contamination are extracting samples at a different time, that is knowns versus evidence, is that
That's right.
As well as witnessing of labeling samples, is that correct?
That's right.
And are those the two primary steps taken as far as RFLP testing is concerned?
That's right.
When you turn to PCR, you utilize those same two precautions, is that right?
Oh, no. No. I've played a video with a horse in the court hearing before, so we can do it again.
utilize those same two precautions and the other
What is a
list of
pony?
precautions that I just talked about.
As far as this case, and you described a little bit about the amplification phase, was it correct that evidence samples in this case were amplified, or did it happen that they were amplified on a different date from known samples?
In this case, it happened that that's the way the testing was done. Not only were the DNA extractions done at a different time, but the amplifications, that is putting the tubes in the thermal cycler and allowing the amplification to take place, that also was done at a different time for the knowns and the evidence samples. That's not necessarily the way every case is worked. It just happened to be the way this case was worked, really based on when we received samples and when we did each one.
Does that play any role in the potential for contamination at all?
Well, it certainly acts as a preventative measure.
But it's not something that you deem to be routinely necessary.
That's right.
As far as these precautions, and let's focus on PCR for the moment, the precautions that you've described that you use in the laboratory, are they unique to forensic science, or are they used in other areas of science involving PCR?
They are definitely not unique to forensic science, because every lab that's using PCR, no matter what the reason, is still doing the same thing. They're taking a small amount of DNA and making millions of copies of it. So any laboratory that uses PCR for any reason has got to be concerned with making sure this amplified DNA that they get at the end doesn't get transferred back to any place along from the start of the sample on forward.
Are these measures taken, for instance, in laboratories diagnosing diseases,
Certainly.
or in research laboratories,
In research laboratories also.
or in laboratories conducting paternity case evaluations or testing?
Exactly. And I don't mean to imply that, for example, a research laboratory would take exactly the same precautions that we would. They may be slightly different, but no matter what the setting is for PCR, you have to be concerned about making sure amplified product doesn't get back to your starting point.
I'm sorry, could I have just a moment, Your Honor? So in terms of these, and just in terms of summarizing these precautions that you take for PCR, they include the way you handle evidence, is that right?
Yes.
The way you extract evidence in terms of when it's done at a same or a different time than known samples in a case.
Yes.
It includes precautions taken with regard to instruments that you use,
Yes.
as well as the direction of flow, so to speak, of a sample going in one direction only, as you described, and not backwards.
That's right.
As well as, and incidentally, are there other steps taken that, to your knowledge, we will discuss later as well, in terms of dealing with the potential problems of contamination?
Yes.
Now, I'd like to shift topics a little bit to specific forensic case samples that are encountered. And in particular, with regard to evidence, can evidence at, for instance, crime scenes be subjected to basically the environment and the elements in that environment?
I don't think it's a question of can. I think it's a question of that's absolutely bound to happen.
Okay, what types of things can happen, for instance, to a piece of evidence found outdoors?
Well, it's going to be exposed to whatever temperatures are outdoors, whatever lighting conditions. There may be a lot of sunlight on that day. Could get rained or snowed on. I mean, whatever, if it's outside, it's going to be exposed to whatever's going on outside from the time that it was left there until the time that it's picked up.
Incidentally, would the same be true, for instance, of a soldier killed in battle out in a battlefield?
I would presume so.
Or dead animals?
Yeah, dead animals too.
You've described a little bit about, for instance, heat. Would that represent one instance of an environmental effect or something that's possible that happens outside?
Sure.
And would that include sunlight as well?
Yes.
And I think you mentioned rain. Or
Well, I
perhaps
mean,
thought-
you said outside, so I was,
Okay.
well, it could get rain. I mean, in California, maybe not, but
Sometimes, some seasons it does. How about humidity? Is that something else that happens?
of course.
Are there other types of influences that are out there outside?
Oh, I think you've named the major ones.
With respect to these influences or effects like heat, humidity, rain, and so forth, what do they do to DNA? What's their effect on DNA?
Basically, all of those things over time will act to gradually degrade the DNA. That is, break it up. The generalization that most everybody is aware of is that dry and cold works to
Humidity, rain, etc., that can actually change DNA from one type to another.
no.
How do you know that?
Well, you know that at a very basic level because these things will not change the DNA sequence. Light, heat, all of these things will not result in changing the DNA sequence. If you think about the kinds of locations that we're testing lengths of DNA. Suppose you're doing an RFLP test and we're using my DNA and I have a DNA fragment that's 5,000 base pairs long. Degradation of DNA is a random process. There's no method or no enzyme that can go in from an environmental perspective and say, we're going to take that 5,000 base pair piece and it will then be converted to a 3,000 base pair piece. Just a nice clean thing, goes from 5,000 to 3,000. That isn't what happens. Degradation is random, so that 5,000 base pair piece, as it degrades, is going to be cut up in many different places. And so it just gradually gets smaller and in many different sizes. So there is no environmental force, there is no environmental effect that can work to simply change one type and make it become another. You may lose the type altogether. You may degrade the DNA so much that you can't type it, but you won't just change types from one to another. It doesn't happen.
Let's talk a little bit about storage. And you mentioned briefly about drying and, making a sample cold as being a means or a method to preserve DNA.
Yes.
Can you describe a little bit more about that? Why is that the case?
Problem with moisture, with high temperature and high humidity, is that enables bacterial growth, and bacterial growth will result in the DNA becoming degraded. That's generally the problem with high temperatures and humidity. Dry conditions and very cold conditions inhibit any kind of bacterial growth, and so they tend to be very good for storage. And keep in mind, unless your sample is sterile, sterile meaning the presence of no bacteria whatsoever, then any moisture and any heat will promote bacterial growth, and that bacterial growth will gradually degrade the sample.
What if you don't, for instance, refrigerate or freeze a particular sample of DNA? What happens?
Well, it sort of depends on what stage it's at. If you're just at the evidence stage where you have some stain on some piece of clothing or some other substrate, then you really will see different effects with moisture and heat. Once your DNA is extracted, and it's really very clean, you don't have bacteria in there anymore. They've been destroyed in the extraction process. You may have bacterial DNA, but you don't have living bacteria. They've been destroyed. And so once your sample of DNA is purified, it does store over time best if it's kept cold. It's usually stored at -70 or -20 degrees centigrade. But if you left it out on the benchtop at room temperature, you could really leave it out there for a very long time before it would be useless.
Your work in the laboratory includes the receipt of paternity samples for paternity tests. Is that right?
Yes, the laboratory's work does.
How do you receive samples for paternity testing?
We
That's the
receive samples
link.
for paternity testing usually by Federal
That's the link to John's new puppy.
Express, and they're usually drawn from the individuals that are going to be tested the day before. They're packed up in a styrofoam container so they can't break. They're shipped at ambient temperature, whatever temperature the air is, and received by us usually about 24 hours later.
And are these in the form of liquid blood in tubes?
Yes, they're usually liquid blood drawn into EDTA tubes.
Are these shipped? Now, you mentioned the term they're stored at ambient temperature, whatever temperature the outside is.
Well, that is, when they're given to Federal Express isn't putting them in a refrigerated vehicle. So they may be flown to us or come in a truck, but whatever temperatures happen to be in that truck or the plane or, all the various things it's going into before it gets delivered to us, that's what temperature they're at.
Does that create any problems in your ability to type these samples?
No, it doesn't.
What happens, for instance, with one of these liquid blood samples? Well, let me rephrase that. Do you, in the course of testing, even in your forensic casework, that is identification casework, other than paternity, do you receive samples in a liquid blood form?
Sometimes.
Are they maintained in any condition as far as refrigeration in terms of their shipment or what they're shipped in?
If we're receiving a liquid blood sample for a forensic case, it's usually shipped in the same manner. Occasionally it's hand delivered. Mostly it's sent by some kind of carrier like Federal Express.
What can happen to these liquid blood samples? What do you see in your casework that can happen as far as the ability to type DNA?
Well, it makes a big difference. If we're talking about liquid blood samples drawn from a known living individual, they're usually drawn in the same way the paternity samples are, shipped to us, and they're just fine. There are other types of blood tubes that you can draw people's blood into that have other types of preservatives other than EDTA. Those other types of preservatives are not as good for maintaining the condition of DNA as EDTA is. That's absolutely that's the preferred way for us to have a blood sample.
Go ahead.
Well, I was just going to say, if the blood samples from someone who's died, then you'd be more concerned to make sure that it had been stored properly and that it had been drawn in an EDTA tube.
Is that because blood taken from dead bodies is subject to this degradation process in a much faster fashion or faster fashion than blood from a living individual?
Yes.
If blood is degraded in a liquid form, whether it's from a sample from a coroner's office or a paternity sample or another sample that you receive in liquid condition, is there anything about that degradation process that can change the types of the sample involved?
No.
What happens when it degrades? Is it the same as happens to, for instance, a stain that you described maybe outdoors?
Yes, it's the same.
Now, as far as other things that exist out in the outside or in the environment, are there any in particular that can create a problem in your being able to type the DNA in that sample itself?
Any sample that is sufficiently degraded can be so degraded that you can't type it by any of the typing methods that are currently available. And there's an exception to that. It's typing of what's called mitochondrial DNA, which is DNA that is not in the nucleus. And that is the absolute last resort. That is, if a sample is in such bad condition, you can't do anything else with it. You might be able to do mitochondrial DNA. And there are not very many places that will do that, that have that capability.
As far as what's out there, so to speak, in the outdoors, what about soil? Does it play any role in the ability to type DNA?
Samples that are retrieved from soil are very difficult to type. Frequently, you do not get any result.
Why is that?
Presumably, you have bacteria in the soil that are participating in degrading the DNA. However, I don't know of a definitive experiment that's shown specifically that it's bacteria. So I only can tell you because this is the common experience. It's in the literature, and many labs have had this experience. Is if they are taking a blood or semen stain from soil, their success rate with those samples is very, very low.
What about leaves? Are leaves another item similar to soil?
Leaves aren't good either.
Would your answer as far as the reason why be the same as soil?
It would.
Is there anything about the presence of soil or leaves that could change the types that are found in a particular DNA sample so that they would be typed differently from what the contributor of that DNA actually is?
No.
As far as laboratory precautions that you take again, and let's focus if we can on PCR typing, the precautions that you described earlier. Is it a danger, for instance, if someone's coughing around a sample? And let's refer to an analyst.
Okay, so what you're asking me is if I have a piece of evidence and I'm going to do PCR analysis on it and somebody's coughing, could that present a problem?
Exactly.
It's possible. It's not, I don't think it's real likely, but I can't tell you that it's impossible.
What about sneezing?
Well, if you make it generic and say, if I, as the analyst, transfer any of my cells onto the sample, then I could create a problem with that sample. But you have to think how much material you're transferring to that sample relative to how much material is there in the sample already. So there's not a black and white answer to that question, and there may not be a simple answer to that question. There is an experiment in the literature where they actually tried to contaminate a sample by the way the sample was handled, and it didn't cause any problem. That doesn't mean that it would never cause a problem. So you do want to be concerned about it, but you shouldn't assume it will cause a problem absolutely every time something like that occurs.
In particular, are you referring to a publication in the scientific literature directly addressing the potential for problems as a result of some of these items we just discussed?
Yes, I am.
Who wrote that publication?
The authors on that publication are Kate Comey and Bruce Bedoli from the FBI.
Could you spell both those last names?
I think Comey is Comey, and Bedoli is Budowle.
And did that publication specifically deal with this question of whether or not these various influences could affect the ability to correctly type DNA samples?
Yes, it did.
What conclusions did they come to?
The conclusion that they came to was that careful, routine handling.
Sustained hearsay.
As far as your reading of that publication, did that lead you to render any conclusions in your own mind about the appropriate way to deal with samples that are obtained, for instance, at crime scenes?
It contributed to my thinking about that, yes.
Did it corroborate what your opinion was before you'd even read the publication?
Well, at the time that I read the publication was during the time we were just starting PCR, so it added to my thinking, which was sort of still being formulated.
Incidentally, as far as a stain, let's say, at a crime scene, to your knowledge, is there any reliable method of determining how old that stain is?
Not to my knowledge.
As far as these environmental influence, actually, let me rephrase that if I may, Your Honor. As far as these items that we discussed, touching, well, sneezing, coughing, etc., do you have a personal opinion about their impact on your ability to properly type DNA samples that have been subjected to PCR amplification?
Yes, I have opinion.
What's that?
Sustained.
With regard to these various impacts, well, let me rephrase that if I can. As far as your own experience in the laboratory with testing samples, and I'm referring to evidence samples, has that included samples obtained from a variety or under a variety of different circumstances?
Of course.
How long have you been engaged in that? How long have you been in the laboratory gaining this type of experience?
Well, when you say you, really we're referring to my whole lab staff. Since before 1992, we did a lot of work. We did a lot of validation work before we actually began to use PCR in casework. So we have the experience from the validation work and from the casework.
As far as in the laboratory, and I'm going to direct your attention in particular to sample handling, for instance, extraction, amplification, and typing of DNA, have you examined the results from your casework since you've used PCR in your laboratory?
Yes.
Do those results include certain steps to determine if, in fact, the process of using PCR and then typing it is leading to or is actually including foreign DNA being typed in samples? Is that question clear?
I think so. There is a
Still an objection, both as to vagueness in the question, and secondly, again, foundation, since we don't know whether it's to samples that are coming in.
It is a speaking objection.
Sorry.
Overruled. Also, comment by counsel not handling the witness. Mr. Clark.
Do you recall the question?
You want to start over?
Sure. As far as this actual PCR typing process that's gone on in your laboratory, do you take steps to detect whether or not there may be DNA present that you're able to determine types from that didn't come from a particular evidence sample?
Yes,
Does that involve the use of controls?
it does.
What is a control?
A control is some kind of sample that you use alongside your other samples or at some point in the process, again, to help you determine whether or not your process has worked correctly and whether or not you have a valid result.
All right. We'll return to the nature of the controls that you utilize in the laboratory a little bit later. But do those controls provide you with an opportunity to determine whether or not foreign DNA, that is DNA that's not part of a sample, is being injected into the process?
They help to answer that question, yes.
All right. And has that gone on since DNA, that is PCR type DNA testing, has gone on in your laboratory?
Yes.
With regard to these potential influences like coughing, sneezing, and touching, do you have an opinion about your ability to detect them as far as if they have any impact on your typing of evidence samples?
Yes.
What's that opinion?
With regard to handling the samples from the point of the extraction forward, the controls will allow you to determine whether anything that you're doing has had an impact on that sample. Obviously, we can't make any determination regarding what's occurred to the sample before it comes in our laboratory, because then the sample is as it is. We don't have any experience in our laboratory of picking up in a sample control the types of the analysts who have handled that sample.
been your experience in your laboratory that these considerations, these things that can happen, simply don't happen in your laboratory?
It's been our experience that we are not detecting them happening in our laboratory. As far as we can tell, they are not impacting on the analysis from the point that we get the sample.
All right, Your Honor, I was going to shift gears again.
All right, ladies and gentlemen, we're going to take our recess for the morning. Please remember all of my admonitions to you. Please don't discuss the case amongst yourselves. Don't form any opinions about the case. Don't conduct any deliberations till the matter has been submitted to you. Do not allow anybody to communicate with you with regard to the case. And we'll stand in recess until 1 o'clock. All right, like counsel here promptly at 1 o'clock.
I'm just trying to think about what the best snacks will be for later. We should have snacks for a live stream. It might be a long one. I don't know how much I'm going to play.
Perhaps the best starting point is that in terms of the mixture results that we have obtained on numerous items in this case, we've never intimated that any of those items match anything. And I know that the court has been concerned with language from legal opinions talking about evidence of a match without a statistic is meaningless. These are not matches. These are results which are consistent with, or in another context, that sources have not been excluded by the testing that was done. One could imagine a scenario where you have two rapists and only one of them is identified, and the victim identifies that person, and both assailants have raped the victim and both have ejaculated, and DNA typing of either form is done, and it's clear that there's a mixture or a combination, more than one sperm donor. We only have one defendant at the end of the table, and his type is consistent with the mixture that was obtained there. It's the people's position in this case, even though that's not directly on point, that that sort of evidence could be presented and explained to the jury what it means and what it doesn't mean without assigning any statistic to it. The court was interested in any legal precedent on the issue. I don't think there's any legal precedent that says that we have to do what the defense wants us to do in this case. And I think that's the appropriate question to ask. What is the legal precedent that forces us to do what they've sought or what they're seeking in this case? There is legal precedent in the conventional serology area that points out that we don't have to do what they've asked us to do. And not coincidentally, one of those cases is a capital case that I tried. I was the prosecutor in People versus Wash, 6 Cal 4th 215. I can speak from personal experience and also relate what's in the opinion, because this is what happened in every sexual assault case where conventional serology was done. Where conventional serology was performed, and we haven't heard much about an aspect of DNA typing that dramatically changed forensic analysis, and that is in DNA typing, it is possible to separate the male DNA from the female DNA in sexual assault cases. And that's why in sexual assault cases, DNA typing of any form is a very powerful tool. Before DNA typing came along, it was impossible to separate out the male biological material from the female biological material. In each and every sexual assault case, the evidence was presented as a failure to exclude. And People versus Wash stands for that proposition. What People versus Wash does not stand for is a proposition that the defense is arguing in this case that has never been applied in conventional serology, that in those sexual assault cases, in order to present any of our results of a failure to exclude, we would have to sum up all of the possible combinations of the biological markers of both the male and the female. And so I point that out because there's a total absence of any legal issue on that point. We were never compelled to do that. We didn't do it in Wash. It was not done in another California Supreme Court case dealing with sexual assault evidence, People versus Ashmus. It is impossible to separate out biological markers. ASHMUS. The site is 54 Cal 3rd 932. So I just point that out because they don't say we don't have to do that, but they do stand for the proposition that the same arguments could have been applied and were not applied in that situation. I think it's important to look at where this issue first came or became injected into this case. I'm not sure who wrote this brief, but it's replete with references to Professor Thompson, where he writes his own brief and then cites things that he's published on this article. or in the article. If you recall, and this began on page 21 and it spanned several pages. If you recall, the argument was first broached in an admissibility context. The evidence would not be admissible unless. And if you recall, the initial argument was that we should not be allowed to report if we had a mixture that clearly came from more than one person, that we should not be allowed to say, aha, we see one of these people in there and the frequency for that one person is 10%. And that was never our intention. Our board does not have frequencies. That's simple. And so the argument
As to mixtures.
as to mixtures.
So
Then
you're only seeking to report that as a failure to exclude.
we're seeking to report the data, and I'll get to that in a minute. Why that's important, and if there are innocent explanations for this, we'd love to hear what they are. But I'll discuss the data in just a second. Professor Thompson's brief then goes on to claim that there's nothing about the forensic context, there's nothing about the observations in these tests that can allow a scientist to assign relative proportions or to sort out what's clearly a mixture. And that's all well and good. That's what Professor Thompson says, but he's not a professor in forensic science. The defense's own primary PCR consultant, the fellow whose name I keep mentioning, Dr. Blake, has published extensively on interpreting mixtures by relative intensities. And I think it's important to keep that in mind. I doubt we'll be seeing him in this case, so this is the most that we can hear from him. But an article, I think it's the casework article in the Journal of Forensic Sciences by Edward Blake, Jennifer Mihalovich, Mihalovich, Russ Higuchi, Sean Walsh, and Henry Ehrlich at page 706. Dr. Blake and his colleagues say, the detection of mixtures depends in part on the fact that any one individual can have at most 2 alleles of a given gene. The presence of more than 2 alleles in a sample indicates a mixture. Even if only 2 alleles are present, a mixture can still be detected if the 2 samples are mixed in different proportions. In this case, a mixture is suspected if the relative dot intensities corresponding to the 2 alleles are very different. And then he goes on to describe other things that appear in sexual assault cases. Later on at page 722 in the same article, Dr. Blake again discusses mixed genotypes. Another issue that has arisen in some cases is the ability to analyze mixed genotypes in biological evidence. And then the passage discusses how you can do that. Probably the most instructive article by Dr. Blake, it's a chapter in a book chapter 17. Unfortunately, I don't have the title of this book. It's called Applications of PCR to the Analysis of Biological Evidence by Cecilia von Beroldingen. That's 2 words, Beroldingen, Edward Blake, Russ Higuchi, George Sensabaugh, and Henry Ehrlich. And in that chapter, Dr. Blake has a very interesting casework example. And I'll just paraphrase it. It's a sexual assault case where the victim had had consensual sex with her husband, I think 9 hours prior. And when the evidence was analyzed, a sample, you always try to obtain a sample from the last consensual sex partner in a sexual assault case, just to make sure that what you're getting is what you think you're getting. And in this case, Dr. Blake not only sorted out by relative intensities and found a much bolder pattern for the person who was accused of the crime, but he said, oh, there's a fainter pattern there, and that's consistent with the husband. And then Dr. Blake, in this mixture, did something that we don't seek to do in this case. He calculated the frequency for the bolder pattern because that was the assailant in the case. Why sum up when you know where the other stuff came from, the DNA pattern from the victim's husband? So while it's easy for whoever wrote that brief to say you can't do that, there's neither the law on their side to say that we can't do. All we're doing is we say, here's a mixture, here are the alleles that we saw, here's the data that we saw with no frequencies. Now, it is, for example, there are 2 series, I think, and specifically with respect to the Department of Justice results. There are 3 stains on the center console of the Bronco. They show a clear mixture. There's more than 2 alleles. And what's clear is that the mixture could not be from Mr. Simpson alone because there's more than one person clearly there. What is also clear is the mixture just happens to be contain alleles from the two victims in this case. We turn the table a little bit when it comes to the glove. The glove, and this, I'm just talking about the PCR context of this glove.
Finally, guys, it finally happened. They finally, and the jury's not even here to hear it. They finally told us after all these months that Nicole and Ron's blood were found inside the Bronco with OJ's blood. Finally, they said it out loud. They said the words. We should celebrate. Like, we should all be doing shots right now.
On the glove, by both RFLP and PCR, there are discrete stains that type very powerfully to Mr. Goldman and nobody else. Very powerfully. The frequencies or the estimates in that are in the millions. There are others which are combinations of both Nicole and Ronald, and those were typed by both PCR and RFLP. And there are relative intensity differences in both the RFLP and PCR results in those cases. And that is all part of the forensic picture on this case. So when it comes to several stains that are right around the notch area where we see these consistencies, we see something that is inconsistent with the two victims in this case that we've proven very powerfully are there. And among the three people that were involved in this case, it is only consistent with Mr. Simpson. And that is all the jury that we want to tell the jury. To preclude us or to try to present some sort of estimate about that calculates a meaningless expression, there is simply no legal basis for it and is totally contrary to the defense's leading expert on this area, whom we will probably never see in this courtroom, Dr. Blake. I think finally, we've gotten the cart before the horse in this case. We have boards. They have launched an objection. I think they're the moving party in that instance, Your Honor, and all they have said is you have to do something. I have demonstrated, and I'll be glad to provide copies of those articles, that there is a scientific basis for us to do what we want to do. They have not demonstrated that there is no scientific basis. I think it would be appropriate at this point. They have never suggested what those frequencies should be in this. And I can tell you, having dealt with statisticians, that you can define a different question that will change the number. I would suggest that, and all we're asking to do is we want to present our results to people who are anxious to hear the results of this typing. The defense does, and there are no statistics on those boards. It has always been our intention with mixtures not to present statistics, but to explain the results fairly, equivocally, and unambiguously, and let the jury decide what they mean. The defense, on the other hand, wants to force us. This is the narrow legal issue that has allowed me to speak for 15 minutes. The defense wants to force us to put a meaningless number up there that they can either elicit on cross-examination. We know they can't call Dr. Blake on this point, but call somebody else to do that. And I think it's time to move on with the trial and deny their objection to this, or the alternative is to force them to sustain their burden of proof that there's both a legal and a technical objection to it. And at that point, we will be happy to explain in or out of the presence to the jury why we should be entitled to present the results in the form that we would like to present them.
Mr. Harmon, when do the people anticipate getting to actually presenting these results?
Could Mr. Clark address that? Because he's up now.
As far as the results boards themselves,
And specifically the mixtures.
I anticipate that we will start the viewing of autoradiographs, but not get as far as the results that are at issue, the mixtures.
This afternoon.
Correct.
All right. Mr. Neufeld.
Well, I think what you do have here, Your Honor, is an attempt, at least by Mr. Harmon, to selectively present statistics to this jury. And frankly, in the words of Marcia Clark, what's really going on here, I think, is an effort on their part to mislead, distort, and confuse the jury by doing so. And in response to Mr. Harmon's suggestion that there's no legal authority to the contrary, there is. People versus Barney in the First Department, they said explicitly that if you want to present DNA evidence, you do so with statistics, otherwise it has no evidentiary value. And Your Honor adopted that same ruling in the court's opinion on the DNA objections on April 7th of 1995. Dr. Blake, by the way, who Mr. Harmon refers to, routinely in trial case after trial case, aggregates in mixtures the various genotypes and always gives a sum. That's been his standard practice. We've seen him do it in more than a dozen cases. And that's a procedure, in fact, used by serologists and geneticists all over the country, as far as we know. It's also the policy and practice adopted unequivocally in the National Academy of Science inquiry into the proper usage of DNA testing, where it's.
Yeah,
I'm going to give it the page. One second. On page 59, it says, and I quote, if a suspect's pattern is found within the mixed pattern, the appropriate frequency to assign such a, quote, match, unquote, is the sum of the frequencies of all genotypes that are contained within that mixed pattern. Okay? So that's what they're saying. I've never seen any scientific authority any place to the contrary. It is the standard practice used by professional serologists and geneticists when
That definitely ain't from me here because I don't use his product.
testifying to the results
No,
of the mixed
no, no.
strain that I'm aware of. I'd also point out, Your Honor, and I don't want to spend much time on it, but you may recall when we brought up this issue last week with you, you said if it had any legal authority to the contrary, that it should be given to the court last Friday and to given to counsel last Friday.
Why did you think
I asked
that?
again whether there was any authority yesterday, and I was told there was none. And all of a sudden, Mr. Harmon comes in here to present his argument, and for the first time, we're hearing references to legal authority. And I don't believe that that's consistent with the instructions that you've given in not only in this particular issue, but all issues that have come before Your Honor during the pendency of this case. I don't even know, because I haven't seen those cases yet, whether or not this specific objection was even raised.
I think this is just a motion anyway. They'll be back to regular programming any minute.
Issue precisely addressed on appeal and resolved by the appellate court. So I don't even know if the cases even stand for the proposition that Mr. Harmon is suggesting. But you do have legal authority here, and the legal authority is Barney. And the conventional serology cases, Your Honor, are not terribly useful in this point because Barney was decided in the context of the power of DNA evidence, a certain cachet that comes with saying you have a DNA match. And we all know that outside this courtroom, that we're talking about things like uniqueness or an extremely rare possibility. We're not talking about items such as 1 in 20 or frequencies of 1 in 40 from conventional serology. We're talking about much, much rarer instances here. And it was because of that that the court in Barney decided that unless you say what that match is or what that inclusion is, and by the way, whether you call it a match or in a mixed stain context and inclusion, you're basically saying the same thing. In fact, Robin Cotton earlier today was asked to address exclusions and inclusions, and it was very clear from what she said that when you're talking about inclusions, you're talking about that so-called match. And I think you even ruled in the past that they're free to use those words interchangeably when describing the concurrence of data in a particular DNA profile. And so what we're saying is, and what the court basically ruled in Barney, is it's a foundation issue. It's a foundation issue. You cannot present evidence of an inclusion or a match, whether it's in a mixed stain or any other type of stain, unless along with it you give the jury some generally accepted view of the statistics of that. Now, you've already ruled that they're allowed to give estimates based on the product rule, and they're not required to use some other approach. But at least they should be required to use that same product rule that they
No, sir, you can't tell them how to present their case. You can give any statistics you want on cross-examination. And then if they don't like it, they will bring up their expert to give different statistics. But you can't tell them how they present their own case.
use for giving frequencies on these single source stains to the mixed stains. And there's a very straightforward way to do that. It's the way that's described in the NRC report. It's the way that any experienced scientist would do it. You simply look at the different frequencies. They could be the frequencies that Cellmark already has for those different genotypes, and you aggregate them. It's as simple as that. Otherwise, you really do mislead the jury. I gave you an example the other day of one mixture that they would present through Cellmark Diagnostics, and that's a mixture on the steering wheel in the Bronco. If you have these other types of evidence where you're saying there's an inclusion and it includes Mr. Simpson or it includes someone else, and you then have a number for that match, and you give it a number of 1 in a million or 1 in a billion or whatever, and then in the very next item, you say we have this mixed stain and it also includes Mr. Simpson, but we don't give a frequency for that. Then under 352, the likelihood of confusing the jury and leading them to infer that that match has the same power as the match just above it is extraordinary. And in the example I gave, Your Honor, the percentage of the population which would not be excluded by that mixed stain profile on the steering wheel is about 38% of the population. Now that's a far cry just on a gut level from the kind of power that we ordinarily attribute to DNA testing. And that's why Barney says you have to give a statistic. That's why the NRC says you have to give a statistic. And frankly, I believe that's why you said it in your decision last month. And I'm simply asking that the same thing be accorded to the people at this time.
All right. Thank you, counsel. All right, Mr. Harmon, I haven't had the opportunity to read the case law that you cited to the court. I will read that, and since we're not going to get to those results this afternoon, I'll have the evening to think about it. All right, let's proceed with the jury. We're going direct examination. by Mr. Clark. Good afternoon, Dr. Cotton.
Good afternoon.
Doctor, you're reminded you were still under oath, and Mr. Clark, you may continue with your direct examination.
Thank you, Your Honor. Good afternoon, ladies and gentlemen. Dr. Cotton, you spoke just before the lunch break about the use of controls, and I believe you briefly described what their purpose is. Could you state again why controls are used in testing?
In general, you're using controls to help ensure at the end of the analysis that the steps worked as they should. That is, an example of that would be you have a type for a PCR analysis and you've typed that sample many times. And so you type that sample along with your evidence samples and you make sure that the type that's expected from this one sample you've typed many times comes out as it should. If it doesn't, it tells you that something could have gone wrong during the analysis procedure.
As far as RFLP testing and the use of controls, if I ask you later to examine and describe an autorad or a series of autorads, will you be able to point out the controls on that autorad?
Yes.
Then let's turn to PCR typing and the use of controls. Do they play the same or in any manner a different role in PCR typing than they do in RFLP typing?
There is one of the controls that's used in PCR that basically plays the same role as the controls in RFLP. And there are two additional controls that are done that sort of play slightly different roles in the PCR procedure.
What role does the use of controls have in this PCR typing process? Why do you use them?
You use one known sample to ensure that the procedure worked as it should. And you use two different negative controls, that is, controls that do not have any DNA in them. And one of them starts at the very beginning of the procedure, and then one of them starts at the point that you set up the amplification. And they are in there to control that in your handling of the evidence and known samples that you didn't add any DNA inadvertently in the processing for the PCR. And there is actually one other type of control that is done with PCR but may not be done in every instance, and that is if you have say a stain on a garment, you might take an unstained portion of that garment and have that go through the process also. That interpretation of that control, should you get something, may be somewhat difficult. And if you don't get anything, then it just says you didn't have anything in this one area that you tested.
All right. The first type of control that you described was a control with a known type of DNA in it for PCR typing.
That's right.
And then you also described, and you used the term negative controls. Does the negative control name come from the fact that there's no DNA in them?
That's right. The known sample that you're typing is called a positive control. That is, you're putting DNA in there and you know what that result should be. The negative control, you are purposefully starting with a blank tube, adding your PCR reagents, but no DNA, and therefore, since you're not adding any DNA, you shouldn't get any result.
And then you also said that there was a fourth type of control?
Well, there's a third type that in our lab is called a reagent blank control. That control is started at the time you're putting your evidence into the tube. You're carrying alongside of that a blank tube. So let's say we have tube 1 and tube number 2, and 1 you're putting your evidence in. In tube number 2, you're not putting anything in there. But then in the process, for example, of doing the DNA extraction, you're adding some things to your evidence. So you are also adding them into the empty tube. So it's a way to make sure that everything that you added to your sample did not have any additional DNA in it.
All right, Dr. Cotton, what I'm going to ask you to do with the court's permission is to, on a single piece of drawing paper, is to describe or list these controls used in PCR typing.
Okay.
Would you be able to do that?
Sure.
All right. Your Honor, may that then be marked as, I believe, 254?
254. We'll entitle this controls.
PCR controls, if that would be
All right.
appropriate.
All right, Dr. Cotton, if you'd oblige us again.
I wonder how many more days of it we have to go through.
Mr. Clark.
Yes, thank you. Dr. Cotton, if you would, could you label this particular piece of paper PCR controls? And I believe you had described that the first control was a control with a known type of DNA in it.
That's the first one that I mentioned.
And I think you used the term positive control?
Yes.
All right, could you write positive control? And if you would describe again what its role is in this typing process.
Positive control contains a known DNA sample that has been typed many times, so the type for this known sample is known. So you have a known DNA sample. You carry that's set up at the point the amplification is set up, and it's carried through the rest of the typing procedure. If the types in that known sample do not come out as they should, it would be an indication that something had gone wrong in that analysis step, in the series of steps that you've used.
In other words, you know what the type of that positive control is, and if you don't detect that type at the actual portion of the test where you determine types, then you know something has gone wrong.
That's right.
All right, what was the second type of control that you described?
The second type I described was a negative control.
And is there one or more than one negative control used in each test?
There is really at least two and possibly a third type, all of which could be considered to be a negative control. But we did give them different names so that you don't get confused.
Okay, then if you've listed under negative control, is there then two versions of a negative control?
Well, it depends if you want to call the substrate control. That's a version of a negative control.
Okay,
Also.
if we could, we'll talk about substrate control separately. So
Okay.
ignoring that for the moment, could you describe the other two types of negative controls used in the test?
Okay, one of them we're just going to call the negative control. And that's set up when the amplification is set up. And the same is true of the positive control. It's set up at the time the amplification is set up. And what I mean by that is that you've extracted your DNA and now you're carrying it into a separate area and you're putting it in a tube and the PCR mix, the polymerase and the primers and so forth. This negative control is set up at that time, and this positive control is set up at that time. And this negative control has everything added to it that all the other samples do, except there's no DNA in there at all.
What do you mean when you say it's set up at the same time? What's that term mean?
Simply that you're putting the things you need out on the bench so you can work with them, and you're physically
Install the old ones on your bike.
going through the process of taking your tube for sample number one, putting in the PCR mix, putting in the DNA. You actually put some oil in these samples also, and then setting that tube aside and going on to the next one.
All right. We made it to the end. Thank you guys for coming out today. I really, really, really appreciate it. I'll see you in about 4 hours. Behave, okay? Be good.